So welcome everyone, and thanks a lot for Glenn and to our speakers today. We will have three people talking about different efforts and different things connected to the fungal conservation efforts. First, we'll have Isabel, then we will have Lucy, and then we will have Gareth, who will be talking about different topics starting from marine or freshwater fungi, and then later continuing with macrofungi and the fungal conservation and red listing. So I think, I don't want to really prolong that. So let's start with Lucy, with Lucy, but Isabel. So I will give you, let's say, the floor, or the speaker part, so we can start with your presentation and you. Thank you everyone for being here.
Thank you. You can hear me?
Yes, we can.
Okay, perfect. Let me see if I can just put the pointer quickly before I start. Okay. Thank you so much for the invitation to be with you this evening to share some of the work that we have been doing, our, let's say, the first steps towards aquatic fungi conservation. And let me start by telling a bit about why aquatic fungi are important. So aquatic fungi are ecologically very diverse organisms. They include decomposers, endophytes, symbionts, predators, parasites, pathogens. They are really, really diverse. And they are very important because they ensure the health, function, and resilience of aquatic ecosystems. Aquatic fungi, they are somehow connected to a lot of ecosystem services from which we benefit. And these include nutrient cycling, these include clean water that all of us need, every organism needs, but also there have been a very large range of metabolites with very interesting properties found from aquatic fungi, as well as genetic resources. So we can think about all the multitude of ecosystem services we benefit from them. Currently, there are more than 6,392 species described, more than 4,000 from freshwater ecosystems, and 2,319 from marine ecosystems. And these numbers are being updated almost on a weekly basis. And if you are curious, I think you should explore a bit these two databases, freshwaterfungi.org and marinefungi.org, because they have a lot of information for these two big groups of fungi. And although they are really important organisms in aquatic ecosystems, aquatic fungi have been really neglected in all major conservation plans and policies. I did this exercise a few weeks ago, and what I did here was I looked at the Web of Science, and I looked for two keywords. I looked for keyword fungi and conservation. And this is a bit the trends on the number of publications. And we can really see that for fungi in general, conservation topic has really picked up in the 90s. And the last few years, it has really gone exponential. As a disclaimer here, this data was actually not curated, and you will understand why I'm saying this. Because when I did the same exercise looking for keywords aquatic fungi and conservation, this is a bit what shows up. So you can see this trend here, which is very shallow. And I'm pretty sure that many of these records here are not really connected to conservation, but somehow the words were together in that particular paper. And what we can see actually is that there is this very large gap in literature focusing on aquatic fungi and conservation compared to the terrestrial counterparts. But also we can see that we are probably about 30 years delayed compared to all the efforts in terms of conservation being developed on the terrestrial fungi. Actually, just very recently, these efforts started to be more visible. And these two papers were really important to highlight the importance of including aquatic fungi into the conversation and in the agenda of conservation. These two papers really started calling attention to, okay, maybe this group of fungi also should be included in these conversations, right? And in the same year, 2022, actually, there was this document that was really important also. This is called Fantastic Freshwater Report, 50 Landmark Species for Conservation. And myself, together with some other colleagues, we were invited to nominate 5 species to be highlighted in this report. And the goal was actually to promote conservation actions for these highlighted species. At the time, and still until nowadays, this is not because these species are vulnerable or endangered somehow, because none of these species have been assessed on their conservation status, but somehow these species are either species that are known to be rare or that only occur in a few locations. And so, and also because they are beautiful. And so these were some of the species that were highlighted on these reports. 1 year after, we were actually very lucky to get this Biodiversity Plus project funded, FunAction. FunAction stands for Aquatic Fungal Biodiversity, Developing Knowledge and Strategies to Inform Conservation Priorities and Measures. And the main goal of FunAction is to actually build the first comprehensive knowledge base on the taxonomy, phylogeny, and functional diversity of aquatic fungi at a pan-European scale. Our project is based on 3 pillars. The foundation, so we want to produce the knowledge base that we need for aquatic fungi-aware conservation actions. Conservation, so we want to translate all this knowledge into conservation planning and monitoring tools for aquatic fungi in close collaboration with stakeholders. And the 3rd pillar is communication. So we are really making a strong effort to increase awareness about aquatic fungi and the role they play in freshwater ecosystems. And just to share with you that this is something that happens very frequently when someone asks me, what do you do? And I tell, well, okay, I'm a researcher and I work with aquatic fungi. And usually the first question people ask me is that, oh, are there fungi in water? So there is still a lot to do in terms of communicating to the general public, but even sometimes within academia about the existence and the importance of aquatic fungi. So one of the key questions that we want to answer within FunAction project is, are existing protected areas effective to conserve aquatic fungi biodiversity and aquatic fungi-driven ecosystem functioning? And this is a question that is closely linked to Target 3 of the Global Biodiversity Framework. Giving a brief summary of the sampling campaigns that we have been doing within FunAction. So we have been sampling in 6 countries, actually a bit more, but in this case, 6 countries from the Mediterranean to the Arctic in 9 different catchments, so 9 different case studies. And we currently have more than 3,000 samples collected for eDNA from water, sediments, and plant litter, for water chemistry, and also for ecosystem functioning. But this is a story to be continued because some of the results are very preliminary and I will not present them here just today. Today, I want to show you a very glimpse on another case study that we have, which is a smaller case study that is not based on eDNA, but is based on morphology. And this case study was developed, it was part of the master thesis from Sergio Costa. And this was developed in Portugal, where I was based until very recently. And in this small case study, we actually collected samples and we analyzed these samples and identified the species based on the morphology of the canidia. And we did this work in 2 different protected regions. So one of them, the Parque Nacional Peneda das Gerês, which is the only national park in Portugal, and another protected area on the more interior north part of the country, which is the Parque Natural Montezinho, which is a nature park. And the preliminary results that we got from this small case study is that actually we have not seen a difference in terms of the number of species inside and outside protected areas. So the protection does not seem to make any difference in terms of the number of species that we observed. It was very interesting for us because this actually, the Montezinho Nature Park was the very first time that we sampled this region. So we don't really have other data to compare, but the protection status does also not seem to be very influential because, you know, the national park have more strict regulations in terms of protection. And actually we found overall lower number of species compared to a nature park that has a bit more flexible in terms of regulation. When we look at the species that actually co-occur in these different areas, inside or outside, we saw that there are species that only occurred inside the park. So for instance, the Peneda das Gerês National Park, we have 29 taxa that only occurred inside, or 12 other taxa that only occur inside the Montezinho Nature Park. But what we found also very interesting is that there is a lot of taxa that only occur outside the protected areas. So we have 5 taxa that only occurred outside the Peneda das Gerês National Park and 22 that only occurred outside the Montsimo Nature Park. And so one of the questions that we want to pursue more here is which kind of threats these taxa might be exposed to, and can we actually do something to protect these species in case of these threats being quite severe. Moving on to a different output from the FunAction project. This is kind of a spin-off of the project. So we are very, very happy with the creation of a new group, a new IUCN Species Survival Commission Aquatic Fungi Specialist Group. The group was created towards the end of 2023, and this was something that was also developed within FunAction, but also was then has grown up to include more experts from different expertise, because within FunAction we are working only in freshwater ecosystems, and actually the founding members also include a lot of experts from marine ecosystems. We were very lucky to have a very strong support from the Global Center for Species Survival. And our group basically has these big umbrellas because it sits under the Fungal Conservation Committee, the Freshwater Conservation Committee, and the Marine Conservation Committee. The vision of our group is actually to develop the scientific and coordination basis for the first conservation assessments, planning, and action efforts for aquatic fungi. I have the privilege to be one of the co-chairs for now, together with my colleague Sally Fryer, which is based in Australia. And currently our Red List Authority Coordinator is Laura Garzoli, based in Italy. We currently have 27 members from 20 countries, spread a bit throughout the world, which is very, very good. We still have a few areas you see in the map that are gray, and we still do not have representatives on these areas. And we are currently taking all the efforts to engage colleagues from these areas that would have the interest and the knowledge to also join the group, to make sure that we have a vast geographical coverage of the group, and we just also have a more complete expertise within the group. Our group has been doing several different kind of activities that kind of summarized here in this slide, between participating in different conferences and expanding the membership, also co-organizing some webinars, and we also have been developing some small projects, and one of them I would like to share with you this evening. This project was actually developed by our member Zephyr Raja, and it was funded by the Indianapolis Zoo in the United States. And one very interesting result from this small project was actually the discovery and the description of a new species, a new freshwater ascomycete, Atromagnispora indianensis, which is very, very good. This was described towards the last summer, actually. And this species also then was one of the species selected to participate in this uproar conservation challenge that was also organized by the Indianapolis Zoo. And it's really, really interesting because then I think it was the first time that an aquatic fungal species participated in this kind of events, and it really brings a lot of attention to freshwater fungal species, which is very, very important. Of course, one of the priority activities for our group is start red listing species. And we are very excited because last month during the Fungal Conservation in Europe conference in Sweden, we started the assessment of this marine species, Digitodispora marina, together with other colleagues. This was very, very important and interesting. It's still ongoing, but we hope that the species will be published this year, the assessments. And while in the conference, actually, I was exchanging with Susanna Cunha, and then she sent me an email, actually, I think it was last week, and she told me, look, I think there are 2 other species that actually have been red listed. And I was looking for more information on these 2 species. So there is actually 2 other species that you see listed here. So one of the species, Limnocaridon incarnatum, is actually an aeroaquatic fungus that has been assessed as least concern. And we have this other species, Aloeloroidis mangrove, that has been assessed as vulnerable. Actually, this species is described from mangrove forests, so it was found in these branches that are submerged constantly by the tides. So both of these species are actually kind of transition species between aquatic and terrestrial ecosystems. So Digitatis spora might be the true, let's say, fully aquatic species, but in any case, this is very exciting times for us to advance red listing for aquatic fungi. Of course, we still have several limitations for the red listing of these species because we have really no comprehensive data on species occurrence, distribution, and geographical range. We still lack information on population size, population trends, generation times, even habitat preferences or needs for many of these species. And so we need still to gather and to keep trying to get more data about these species and their ecology and then distribution to be able to then also perform the red list assessments. And knowing these gaps exist, we actually are working on trying to fill these gaps. So one other project that we have now currently ongoing that started in 2024 is the Most Fun project. And Most Fun stands for Monitoring Strategies and Tools to Address Knowledge Gaps on Aquatic Fungal Biodiversity. And the main objective of the project is to develop knowledge, approaches, and network needed to initiate routine monitoring of aquatic fungi biodiversity across Europe. I will show you some of the objectives that we have within the project. So one of the objectives we have is actually to collect and reanalyze published data sets and metagenomes to extract data about aquatic fungi. So this comes from databases like GBIF, Magnify, Global Fungi, and others. One other objective that we have within the project is to reanalyze DNA from other biodiversity monitoring programs. So DNA or even bulk samples that still need to be re-extracted, but the idea here is to make use of samples or DNA that was collected for other purposes, for other organisms, and the information is just sitting there and waiting for us to extract this valuable information for aquatic fungi, and this is one of the big objectives of our project. Of course, there are so many environments that have not been sampled yet. So one of the objectives that we also have in our project is to perform novel sampling in understudied environments. This is, for instance, glacier and estuaries. And one thing that we believe it's very important is that if we want aquatic fungi to be included into monitoring programs, methods need to be very standardized. So one of the things that we want to develop in our project is these guidelines for monitoring. And one of the tools that we have is essential biodiversity variables. And so if you have interest in this topic, we are organizing a workshop next month during the World Biodiversity Forum in Davos in Switzerland. So if you are coming to the conference and you have an interest, we still have a few seats available in the workshop. So just screen the QR code and or get in touch with me directly, and I can give you some more information about that. Of course, we believe that all the efforts that we are developing right now should not just end with the project. So one of the things that we want to do is to make sure that this coordinates stakeholder engagement that does not stop with the project. So we want to build a knowledge to action hub on aquatic fungi biodiversity to make sure that the works that we start now will continue in the future, and we keep the momentum and the critical mass to really make sure that we make strong advancements towards including aquatic fungi in biodiversity monitoring. And I will very quickly give you a glimpse on some results that we have on the first objective, which is collect and reanalyze published datasets and metagenomes. And this is a work that is being done by Red Calori, which is a PhD student within Most Fun project. And Red is currently compiling information of the distribution of aquatic hyphomycete species from a multitude of repositories. Here you see the map for the data collected from GBIF, Global Fungi, and PlutoF. And you can actually see that in some parts we really have an overlap, but in many parts of the map we really gain a lot of information from compiling information from all these databases into one single database. And one of the things that then we can do with this information is we can zoom in into particular species. So this will be very important, for instance, for red listing of species. And just as an example here, this is the map of the current map of distribution for Elatospora cuminata. And this is information coming both from morphology and from DNA. There are currently more observations actually coming from molecular data than from morphology data. And there have been records for more than 700 sites. One very interesting curiosity that we notice here is that, for instance, Portugal is empty and Brazil is empty. And I know that there is data available for Alantospora combinata from Portugal because I found the species multiple times, and this information exists in publications. And also from Brazil, I received this information from one of our collaborators. And so one of the things that we want to do also within the project is to make sure that we also compile data from the literature to make this database even more comprehensive. So very quickly to summarize our future steps. So we believe that it's really important to create this baseline of aquatic fungi occurrence and distribution data. This is really important, even just to start a lot of open questions that will be answered, but also it will open even more questions. So also one thing that we believe is very important is to understand how aquatic fungi respond to environmental changes. And both these questions are very important to then perform red list assessments. So we need to know who is there, where they are, what kind of threats they are suffering from, can they adapt or not, and then perform the red list assessments. Of course, we believe that it's really important to incorporate aquatic fungi in conservation planning and action, and we believe that we will be able to do at least some pilot studies on conservation planning within the FunAction project. And one thing that we believe is very important also is to keep raising awareness about aquatic fungi and their importance. And we have been developing a lot of activities on that. And maybe some of you that have met us in Sweden probably have a sticker that we developed within the project for this. And last but not least, training the next cohort of experts on aquatic fungi. This is really, really important because we need to keep the knowledge growing. The community is still rather small compared to for instance, to the terrestrial fungi colleagues. And so if you have curiosity, or if you want to know more, or if you want to learn, just get in touch with us, because we would be very happy to organize workshops or webinars to train, to share with you protocols. Just get in touch with us. And just to some acknowledgements, because this work is of course not done. I did not do this by myself, of course. This is the list of co-authors and the collaborators, either from FunAction, Most Fun, or from the specialist group, and of course the funding agencies. And yeah, that's it. Thank you so much.
Thank you very much. Thanks a lot. And I think we will have questions in the end of the whole 3 seminars. And because I also have some questions, but thank you anyway for great talk, and we will come back to you. And now we will have Lucy representing Czech Mycological Association, or it's somehow different name of the association.
Yeah, it's Czech Scientific
Association for Mycology, something like that.
Yeah, yes. Yeah. We have two associations for some reason. So.
Okay, so I think we will now have Lucy, and then we will come back to the questions to Isabel. So thank you very much, and if you have time, please stay with us. Oh, so, perfect. It's working. Yeah. Great.
Okay. Well, so we Lucia Ivanova, and we will a little bit change the scale, because we will go more local, and also the fungi I will talk about are a little bit bigger, because I will talk about how we made Czech, the new Czech red list of micromycetes. That means those fungi that are visible to the naked eye. So we move back to the end of the year 2022, at the time before we started our work on the new checklist, new red list, and at the time, and we still have 46 protected species in the Czech Republic, and this list is pretty old. It was composed at the end of the 80s, and some of the species in the list of the protected species, we really don't know what it is, and some of the species are were shown to be pretty common, so no change since then. Also, legally, all the fungi are plants, and there is no change coming. So, sorry. The first red list was published in 2006, and it wasn't based on the IUCN criteria. It used IUCN terminology, but it was based mostly on expert evaluations. So the red list was pretty good, but it after so many years it was a little bit out of date. So we wanted to make a new one, but we wanted. We were still waiting for some big grant to finance all this big great red list with all the field work and data collection in the right way. But this grant wasn't coming. So at the end of the 2022, we were approached by the people from the National Conservation Agency, and they offered us they will provide some limited financing for the new red list. But the red list needed to be finished within 1 year, and we need to use IUCN guidelines for real this time. And because of this short time limit, we won't gather any new data, so no field work. So it would be easy just to assess the available data. But you haven't got any national checklist. You haven't got any central database. All the data was scattered through thousands of sources. We don't have any prior experience with IUCN guidelines. In fact, there is, I think we were the first Central European country that actually used those IUCN guidelines. We haven't got any specialists, we haven't got any specialists on Entoloma, Inocybe, Thalamonia, because the taxonomy is evolving so fast. So even we have an expert on Inocybe, he just said, well, I'm finished. I won't work with Inocybe anymore. And yeah, the time was limited. So we devised a 5-phase plan to develop the red lists. We started with species suggestion because we haven't got a checklist. So everybody from the mycological community could suggest some species that they think would be worthwhile to evaluate, because we didn't want to evaluate very common species such as Amanita muscaria, because we needed to save resources. The species suggestions was made in open Google tab, and everybody could suggest species and write some reasons why they suggest those species. Then we gathered all the data and put it on the one big heap, one big spreadsheet, and based on those gathered data, we then assessed all the species, and after that, we as the editor team then reviewed the data and submitted to the National Conservation Agency at the end of the year 2023. The next year, we just wrote the accompanying manuscript, and the red list was published at the end of the 2024 in Shiroda Journal. Something about data. We use data from various sources. We have National Protection Database. Both of our mycological societies have their own database. Some of the databases use public and private collection from herbaria, reports, notebooks, and so on. those data because they weren't collected specifically for the red list. They have plenty of issues. There was a huge bias towards protected areas, and we haven't got much data from the regular countryside. Also, there was a huge bias towards already red-listed species and protected species. A lot of the records have poor equalization. There were a lot of taxonomical issues. There was some sort of mushroom picker mentality, when some of the people weren't actually very keen on sharing the data. And also a huge issue was data inflation in the past 2 decades due to some projects and some applications like iNaturalist and so on. So it was really hard to infer any trends in the data. The red list is based only on spore caps. We didn't use metabarcoding data, at least not systematically, and also we omitted Hippogynous fungi and those very tiny ascomycetes because it's very complicated to get some sensible distribution data. We have gathered about 70,000 data points. It doesn't seem it's too much, but it's only for those species that we suppose that are rare or endangered. So if we gathered all the data from the Amanita muscaria, there would be many more data. The data was evenly split between already existing databases, data from individual mycologists, and from Fungaria. And at the bottom right, you can see how the chart, how serious is the data inflation in past two decades. Its number of records, and you can see since 2005, the inclination of the chart is really steep. We covered most of the country, but the coverage is very uneven. You can see a lot of data came from around those 2 major cities, Praha and Brno, and also from those nature landscape areas and national parks. This is on logarithmic scale. The assessment was carried by 20 experts, both amateur and professional mycologists, and they were using a web application developed by Monika Koleniova. And since we have this web app, we still need those experts to look at the data and how to interpret those data, because if only those people with the field and taxonomical experience could say if those data have any meanings. We also stress the higher resulting category would be the more thorough review of the available data. Should be. We put all the results in a shared Google table, and then the results were reviewed by the editors. We have some problem with IUCN guidelines. There was some pushback against the usage of the IUCN guidelines, because some people thought that they were too complicated, and some were saying we are comparing apples to oranges. We are comparing, Amanita muscaria or Rhodotus palmatus to those white rhinoceros. But that's what red listing is actually about. You need to compare those apples to oranges. And yeah, we need those experts, so we have to a little bit simplify some things, to choose some proxy data, because we have trouble with those trends. So we look on the trends, like, for example, how does the elm tree decline, and we can base some at least rough trends from this proxy data. So yeah, so the lesson is, do not try to over-explain those Bayesian criteria, more focus on the use in the practice. Well, some results. We evaluated over 1,800 species, and of those 1,600 were evaluated as threatened or data deficient. The red list is heavily biased toward basidiomycetes, but it's expected outcome because those basidiomycetes are mostly represented by the pasicle macromycetes. We use mostly C criterion, and you can see below that the average category decreased by 1. It's not because the threat to the fungi decreased, but it's because Bayesian criteria is more strict in those high categories, critical endangered, more than those criteria that were used in the previous red list. Also, we have much more knowledge about distribution and ecology of the species, and in some species there was actually a reduction of the spread, like Flebia or Masonia centrifuga, that spreads currently in the Czech Republic very, very significantly. The good result is that we found over 50 of 80 possibly extinct species, and some of them actually turned to be pretty common, like Arhenia artioluga, Typhola quisquiliaris, Polyzeus atrophuscus, were actually moved from the possibly extinct to the least common category, so no threat to them. There is a chart that illustrates how the categories between those two editions of the red list have changed, and you can see that we assign category to most of the data deficient species from the previous red list, and on the other hand, almost half of the newly suggested species are now evaluated as data deficient, but we hope in the next edition they will be given a proper category. So, something about the future. The next edition is scheduled from 2034. We really wish this time to have a central database, because the data handling was the most time-consuming part of the red listing. We also wish to connect the database to the GBIF, and possibly we can also use it for some automatic generation of the checklist. We also would need to bear with the prominence of the meta-barcoding data. What is underway is now the revision of the list of the protected species, but since the political situation here towards conservation isn't very good, I put much hope into the revision. So thank you for your patience, and that's all. Marta, you are...
Yes, thank you, Lucy. Marta, you are still muted.
Okay, thanks a lot for telling me this. I just wanted to say that thanks, Lucia, for presentation. And after Gareth, we will have a discussion. So if you have some comments, you can already post them in the chat. But in the end, we will have a proper room for discussing and asking questions. So yes, Gareth, I think you can share your screen and we can move on to the grasslands and macrofungi. Thanks again.
Does that work?
Yes, it's working.
Brilliant. Well, first of all, thank you to Glenn and Marta for the opportunity to speak to this webinar. And I should point out where I'm based, which is where a lot of our field work is done, is on the far west coast of Wales. So the sea is just there, maybe a few hundred meters. And I was going to talk about how we've deployed eDNA metabarcoding to assess the biodiversity value of fungi in ancient grassland habitats. In case I forget at the end, I need to recognize that this is not me only. It's Andrew Detheridge, Louis Tranter-Ruby-Bai and Lizzie Wary in my group. And we also work a lot with citizen scientists like David Harris and Emma Williams, and also in Natural England, Matt Wainhouse. As we're in Wales, our statutory body for the environment is Natural Resources Wales, but they don't have so much money at the moment. So Natural England have been brilliant for us, and Martin Ainsworth at Kew is always brilliant to talk to about taxonomy. So first I need to tell you a bit what I think an ancient grassland is. I think maybe this is a term you haven't heard before. Eve Arnolds, who was the first person to highlight the importance of sort of these particular grassland habitats for a suite of grassland fungi, including the wax caps, a Hygocybe species, Hygophoracea. He mentioned the term wax cap grasslands, and he estimated in the 1980s there was maybe only 200 hectare left in all of Netherlands. So that's a really serious decline, especially in lowland areas. More than 95%, probably higher than that, has been lost to mechanized agriculture, ploughing, and then also diminished further by the use of synthetic fertilizers. So that's why these habitats are particularly rare, and we would like to be able to identify and protect the remaining habitats. Sometimes they're hard to identify because if you have heavy sheep or cattle grazing, that can remove the plant diversity. So a plant ecologist coming along doesn't recognize that the soil is more or less intact and contains valuable biodiversity. So typically we're struggling, or we're working towards trying to find a date to say, how old is ancient? In the British Isles, they use the term ancient woodland. So we'd like to have something similar to that was a simple definition. And we're not even sure at the moment. These are early days, but we think maybe 80 years as a minimum, and it's not really feasible from historical data to go beyond 200 years. And within this definition of ancient grasslands, it seems that the grassland fungi are very useful indicators because they seem to be very long-lived and take a long time to develop from spore to fruit body, and maybe last, maybe longer lifespan than a human. The term I'll use, it's a very ugly word for which I apologize. We use this term CHEG-D, the Claveriaceae, Hygophoracea, Entelomataceae, Geoglossacea, and Dermaloma species, as the term for the suite of species we find in these habitats. So if you look at this view not far from here, you can see here is an ancient grassland in the foreground, and further back you have these fields that most members of the public would say that's a field that looks like similar type of grassland, but that is sorry, someone's trying to get in. Those fields have been ploughed and reseeded with ryegrass and have very low biodiversity value. They're essentially like a crop. in addressing how rare these grassland fungi are, there are quite a few species of mostly wax caps that are assessed as globally vulnerable. And to be able to communicate to non-mycologists what that means, that's a similar conservation or extinction threat to that faced by the great panda and the giant panda and the snow leopard. But just like the panda and the leopard, the grassland fungi are threatened by habitat loss. So the method we use is eDNA meta-barcoding. And we use these devices like this HiSeq, MySeq, to mass sequence all the ITS barcodes amplified from a given soil sample. So you maybe get 50, 100,000 species from one sample. And the more abundant species that are sequenced more times, so you kind of get a species list and the relative abundance, which ones are more abundant than others in the sample you're looking at. So I thought I'd take you to a field site that's quite close to here, maybe 15 kilometers inland from us. It's an upland grassland restoration experiment. This field was last ploughed in 1970, so it's not particularly special for diversity of wax caps, but they are recolonizing. And we were keen to look at exactly the factors that were affecting recolonization. And here you can see there's 7 treatments. There are these paddocks, maybe 30 by 30 meters. Some are grazed by sheep, others are hay cut. Here's the regime of summer grazing with sheep, hay cutting, and half of those hay cut plots are grazed afterwards by sheep, and some are limed. And this first treatment is what my farming colleagues call the control, but it's not the control, it's the human input standard farming treatment, where they add some fertilizer, some lime, and they graze the summer. So we've been looking at these plots for maybe 25 years, and we used to do annual fruit body surveys, and you can see that most of the fruit bodies are on the hay aftermath grazing and lime or not, just because the sward is short in the autumn. The mushrooms don't come up if the sward is long. But you see a slightly different picture if you do the eDNA metabarcoding. And the first thing to notice is that in several of these plots, the Cheg-D fungi comprise maybe 50% of all the fungal biomass in the soil. So they are dominant components. Whereas in the fertilizer plot, you can see it's much lower here. And by looking at the fungal communities, you can see that the fertilized plot is quite distinct from the others, and the others are also separated by the addition of lime or not. So the process we evolved for the eDNA metabarcoding was to take about a 30 by 30 quadrat and take 36 cores on a grid in the quadrat. So you end up with a bag of cores. And this bag sort of represents the plot or the area. And we freeze dry this, we grind it finely, and extract the small amount of DNA, extract the DNA from a small subsample. So really we're trying to subsample efficiently or effectively the whole area in this maybe one kilo bag of cores, and then homogenizing this to take the 200 milligrams from that. And after extraction DNA, we amplify and sequence the ITS2 barcode locus. So you have lots of sequences, but the harder part is, that's fairly straightforward technology, the eDNA, the meta-barcoding part. The harder part is to associate accurately the correct name to the sequence. And we make great use of this fantastic Estonian invention, Unite, which I'm sure others of you use. And here, Urmas Kolliag and his colleagues have created the term species hypothesis, which is for a cluster of related species. And they focus more on the number associated with the species hypothesis, which stays the same, even if the Latin binomial name changes after some taxonomic reassessment. So I'll give you an example of a rare grassland fungus where this has happened recently. This is the violet coral, Clavaria. It used to be called Clavaria zolingeri, and there was a taxonomic reappraisal of it by Olariaga et al. In 2025. And they found that commonly across Europe, there are 2 species, and elsewhere there are other species. So you see here that the species hypothesis number stays the same, but the Latin binomial name changes. And the species that we find that we used to call Clavaria zolingeri in Europe turns out to be 2 species, Clavaria violacea pulchra, and also the more distantly related Clavaria amethystina. So this is where Unite is brilliant, because the number stays the same even if the Latin name changes. Unfortunately, it's very pleasing that even though this has been split from one species into two, if you take your microscope out and examine the spores, you see clear differences between them. And having discovered this, we now find across Wales that we have both species, and I think that's the case for many European countries. We also do some sort of taxonomy, or defining new species. Here's one species we recently named a few months ago, Gliophorus albovicidus, which is it's very similar to Gliophorus irrigatus, but just a paler color. As you can see, Gliophorus irrigatus is a darker shade of brown. And we assessed using global fungi the distribution, and we find that it's got a broader Eurasian distribution, similar to Gliophorus irrigatus, but it's still very rare. I guess in the fullness of time, a red list assessment would be done of this. We've only found the fruit body in 2 places, but we also have found it in 1 place via the eDNA only. So you can see here's the purple, here's our new species, here's the standard, the more familiar Gliophorus irrigatus, and because of the eDNA databases and iNaturalist, we could see there were lots of other species in this complex, so we also named them with the help of some American colleagues. So we ended up naming more American species than European in this study. So now I'm going to take you back a little bit to, I guess, why I still do this work, or I started maybe 25 years ago, and there was a big court case 20 years ago in Cardiff, the capital of Wales, and it was to do with the redevelopment of this reservoir. And you can see that the grassy embankment around here was home to 28 species of waxcap. So the statutory body in Wales, it was called the Countryside Council for Wales, notified this as a site of special scientific interest to give it legal protection. But the owners of the reservoir were very unhappy about this, because they wanted to build millions of pounds worth of houses. So they went to a court to challenge the legal decision. Unfortunately, it went to the highest court in London, and the waxcaps won. I think if it hadn't won, I would be doing something else now. So this was a landmark case. But it's interesting to revisit the same place and see, what do we find with the eDNA? Do we find similar results to the fruit body surveys that were done 20 years ago and more recently as well? And we do. So you can see this cluster of these are the sites with the biggest diversity of fruit bodies, and they're also ordinating similarly in terms of the eDNA. So this type of stuff is important. This type of ground truth thing, they call it, is important, just to see, does the eDNA tell you a similar story to what fruit body surveys would tell you? And a few years later, we were approached by some people in Birmingham. This is the 2nd biggest city in England, and they were looking in the middle. This is not a promising area to go to look for unusual biodiversity. It's maybe 5 kilometers from the middle of Birmingham, and Birmingham is where the global industrial revolution started. So you wouldn't think it was promising, but because of the steep hillside here, there were lots of waxcaps here, because this land had never been built upon and it hadn't been ploughed or anything. And we looked at these areas with the eDNA. The people here in the picture had been previously looking at waxcaps since the 1990s, and they had found 22 species. But with our eDNA work, we were able to find 6 more, including some of these species. 3 of these species are now assessed as globally vulnerable. So this increased the strength of the case to provide legal protection for this site, which has now happened in 2019. So I think this is the first time we've used eDNA evidence to help achieve global legal protection for a wildlife site in any group of biota. So eDNA evidence isn't admissible really in biodiversity assessments, because IUCN criteria talk about mature individuals. So what we did here was we said, Microglossum olivaceum is to be found, its mycelium is in this quadrat. So if you look very carefully, the next autumn season, you might find it. And this is what happened with these 4 species. When we told them where to look and what to look for, it was easier to find them. So typically, we obtain something like, this species list here, and these are showing the relative abundance of those particular species. This is the mean here. So you see the sort of rarer species and the more common species that are found. We find some unusual things relative to fruit body recording. For example, Angustifolius is possibly a new UK record. Flaviposoides is a new species defined from Norway, which also seems to occur in England. And this is the most strange one. Gliophorus subaromaticus is a white-coloured relative of Gliophorus irrigatus, but unlike the species we found, it's got a distinct burnt plastic smell. And so far, it's only known from the redwood forests of California. So we're kind of a little bit puzzled why we find it here. We're certain this is the same thing from its ITS sequence, but we're still rather puzzled. This may be something to do, maybe it's just a single spore landed here in the Peak District, which is in the middle of England, and has not found a mate. This is what we think is the likeliest explanation, but it's still quite puzzling. So you do have some differences between eDNA data and fruit body data. We're still working, our main focus now is on defining ancient grasslands, mostly through my 2 PhD students. There's Bradley Powell, who's looking at the stratification of the soils. Jake Dalzell is looking at mycoparasitic fungi that are associated with the plants in these habitats. And Ruby Bai is looking at soil biota, soil animals in these habitats. We wrote a report about the different criteria you might choose to define an ancient grassland, which you can find here. And we think we're just becoming more convinced that the Chegdi fungi are kind of quite good indicators, better than any of the plant species, for identifying candidate ancient grasslands. And I'll just give you one example before I finish. I just need to look at my clock to make sure I have 3 minutes. Here is the world's oldest agricultural experiment, and this again is near London in England, and it's park grass. It was started in 1856. And the aim of it was to look at what happened if you add a different type of fertilizer to hay meadow productivity. It was known that this field hadn't been ploughed since 1815 at the end of the Napoleonic Wars. And in 1874, there was a single fungal fruit body survey done. And it must have been done well because the Reverend Miles Berkeley, who founded the British Mycological Society, checked the IDs of the species. And there were several Chegdi fungi present here, and I'll take your attention to what's now Hygocybe coccinea, is a bright red wax cap. I always tell people, if you know nothing about wax caps, just look for the red ones. The red and crimson ones are the ones that are the most slow to establish in a grassland habitat. When we went there 20 years ago, we found large numbers of Hygocybe punicea, this crimson species here. We found them here and here only, and these are the only 2 plots on this whole field that didn't receive any additions of any fertilizer. So you can see how the eutrophication of the soil, especially with synthetic fertilizers, is harmful to these fungi. So I just have a map there to show the distribution of the fruit bodies, and also how the eDNA and the fruit body survey data are consistent here as well. And finally, I know Glenn was keen on this, was the what do you do if you have an ancient grassland that has lots of these Chegdi fungi on it and it's threatened? In this case, in this set of pictures here, it was a major road in South Wales that was going to be built. There was no way they were going to move the road for the fungi, so the only thing to do, and it was done in very short notice, was to move the fungi, or to try to move the fungi. So you can see how the digger driver here, he basically moved the turf maybe 50 meters to the south. And here's his final work here. So I would say that not all heroes wear capes. This guy was heroic in what he managed to do in a short period of time. And there is a video that you could find if you put wax cap translocation that shows another translocation that was more ambitious for building a hospital in Ipswich, which is in the east of England. The turfs were all moved maybe 40 kilometers to a new site. And I see Marta's getting slightly worried that the time is running out, so I'll stop there. And thank you very much for your attention.
Thank you, Gareth. It was really nice to see again. Yeah.
Thank
so
you. Thank you
to
very
our
much.
to our three speakers. Maybe just that to be completely clear, these three people are not really part directly of FunDive, but this kind of conservation-inspired meeting came out of the ECCF, the European Conservation getting together of mycologists that happened in Uppsala just over a month ago. And what happened is that Marta and me, we were so amazed by some of these talks, three of which you have seen today, that we really wanted them shared again with a broader audience. So I do thank the speakers again. So now I imagine that there will be questions. So please raise your hand if you do have any questions. I will momentarily allow all of you to unmute yourselves and also to show your videos. And then Marta will lead the discussion, I guess.
Yeah, I think we have already some raised hand, if I'm not mistaken. Yes, it's Alfred. So if you want to ask a question, you can do that now, I think.
Hello, can you hear me?
Yes, we can.
Okay, well, thank you very much for all the speakers. It was really interesting talks. I had a question for Gareth. So up here in Scotland, there's a huge amount of afforestation happening on waxcap and unimproved grasslands. A lot of the policy debate is centered around carbon, and carbon in the soil is not as big a factor as people are often looking at carbon stored in trees and stuff. But is there any way you think that wax caps could be used as a sort of proxy for soils that store a high amount of carbon? And then could that be a way that wax caps are used, or wax cap fields are sort of protected from a carbon storage perspective, which would be actually quite relevant up here in Scotland?
You know, I think it's a bit sad that most of the politicians and the members of the public think that all the carbon is in the vegetation you see above the ground, whereas more than 2/3 is below the ground in the soil. So these ancient grasslands have a high content of carbon. So the benefit of afforestation on those things is minimal and needs to be weighed against the loss of biodiversity. And also, it's not always the case you plant trees and you can increase carbon sequestration. I think if you were to do it on some of these grasslands, the drying of the soil by the tree roots could lead to net carbon loss from the habitat. So they do need to be more careful and actually pick not destroy biodiversity in the aim of a limited amount of carbon sequestration. It's the same in Wales. And I think maybe in other countries in Europe where tree planting is heavily promoted these days. We need more trees, but in the right place.
Sorry, can I just, I probably didn't frame it properly. But yeah, I'm definitely aware of that, in that it releases all the soil organic carbon. I was just trying to see, is there some way that you think that wax caps could be put into the policy arena as this method of, or a sort of, as showing that these grasslands are good in their own right, plus they're also indicating that carbon's being stored in a way that can maybe get through to the public in that sort of sense?
Yeah, I think so. Especially, I just tell them to look for red wax caps. These would be an ancient grassland, and they should then draw somebody's attention to it. And if there's a plan to plant trees on it, they need to do a fuller assessment and not assume that they're going to do necessarily be beneficial in what they do. Yeah, so just tell them to look for red wax caps. Try to simplify it, because a lot of the public find taxonomy of any sort difficult.
Thank you.
You're welcome.
Thank you very much for asking question. I don't see any raised hand, but it's still a time. Oh, Jakob, Jakob is having a question just now. Jakob, can you hear? Oh, yes, you're unmuted now. So, Jakob, can you ask your question now? If I can't hear you, maybe others can. Oh, now we see you.
Yeah, we still cannot hear you, Jakob.
Oh, not yet.
Can you hear me now?
Yes.
That's better. I was muted also on my computer. So thanks for the nice presentations. I was curious first to ask Isabel on, I mean, it seems to be a really long shot to go into a red listing process for aquatic fungi since they are so poorly known overall. I mean, even for terrestrial fungi, it is a long shot because we compared to birds or mammals and plants, we have really limited knowledge on the global scale. But it seems that you still in your projects use this as a driver of compiling data, or is it rather the other way around, that you do the red listing because you're compiling the data? I'm just curious to the incentives in the work you're doing. What is the driver of the work you're doing?
I think it's a bit both ways. I think compiling the data came first. So, when we started with the project Fun Action, the main idea is to start some kind of actions towards conservation of aquatic fungi. And of course, red listing would be one of the obvious first steps, because this is something that any stakeholder, policymaker will ask us. So, if we want to have included fungi or aquatic fungi into conservation planning, one of the criteria they will probably ask us is that, why is this species important? And then one of the things we can tell them is like, okay, this species has been assessed and it's vulnerable or it's not, right? So for us, one of the things we wanted to do, of course, is to start having the knowledge about the species distribution. There is so many questions that we cannot answer currently because we don't have yet enough knowledge about that. And this is one of the things that we have been doing within Fun Action. So we have sampled these case studies, but also we teamed up with our colleagues from Estonia that were already having a project ongoing called Fun Aqua. And in this project, there is, I think the first data paper will be out very soon, because we just received back the comments from the reviewers. And in this one, we have more than 1,000 locations globally that have been sampled for water and sediment. So this will build up already some information about distribution of species. And of course, we have some information based on morphology, but this is very limited because basically, well, this happens, I think, to most people. People sample in the surroundings or the places where they are established, right? So then there is so many locations in the world that have not been sampled yet. So I guess answering your question, we started to really try to invest into getting data about species. And of course, we want to maximize this data to also try to do for the species possible, try to start the first red list assessments.
Super, thank you so much. Then I have a quick question to Lucy. I'm just curious. I know that that data from the Czech Republic are not. I mean, you've done a really big job in compiling the data from all the different data sources, but I know that there's still a recording issue in Czechia in terms of sharing data. Do you? So my question is whether you believe that this process of the red listing has been helpful in securing that in the future some of the important data on fungal occurrences in the Czech Republic will be more likely to be shared openly on the global scale? I mean, in GBIF and other sources?
Yeah, absolutely. We have overcome a huge distrust of data sharing But nowadays, I think more people are open to share their data. Well, to share the data within the country, and also we are slowly trying to persuade people from the institutions to share the data to GBIF, because if you see a lot of studies that use some worldwide or Europe-wide data, Czech Republic is always missing, because there is very little data on GBIF from Czechia. So hopefully, fingers crossed, so we will get the data there eventually, but it won't be tomorrow. But within a few years, it's possible.
Sounds amazing. Thank you.
Thank you. We have one more question and raise hand from James. So James, you can ask your question now if you're still there with us.
Hello. It was a question for Gareth. I was wondering if you'd had any results from the turf translocations, whether there's been any fruiting bodies or if you've done any eDNA sampling on them. Did they actually work?
You are
Gareth?
muted.
Oh, you're unmuted. Yes.
Oh, that was a question for Gareth.
Yeah, sorry. So it's me that was being incompetent here. Sorry.
All right. Okay. Sorry.
Yeah. We did sampling before, and then we did sampling afterwards. And I think it was 9 months after translocation, and we have a second set that we're doing at the moment. So it didn't seem anything drastic had happened, but I think maybe I'll be dead before we really know if it worked in the long term. It was quite weird that one of the species we translocated was that the new species we discovered that we only found in 2 other places. So that seems to have survived, and it seems to be the relative abundance is a little bit higher now, but I don't read too much into that. It was just more rather strange that we accidentally moved a new species. That was a bit weird. Yeah, but I think that they have new laws in England, biodiversity net gain, and it requires long-term monitoring. So we have to do it, I think, maybe next year, and then in 2 years, and then in 5 years, and so forth, to see the long-term effect of that translocation.
Thank you. I see we have one more question from Bruder. So you can ask if you... Yeah.
Thank you. Can you hear me okay with this?
Yes, we can.
Also to Gareth, a follow-up question, maybe to the previous one. And I was wondering with your turf translocation study, who paid the driver of that digger there? The question is, of course, do you get funding for as a compensation, for instance, for this hospital being built or the road being built? Is there a scheme that obliges these construction projects to compensate and therefore to have some kind of restoration measure funded as directly through the project?
Yes,
the construction project.
Yeah, I think that project with the hospital was very expensive. I imagine, million euro or something. I don't know. It was quite expensive, but that's part of this new law that's in England. I'm not quite sure what's happening in Scotland, but because environment is devolved power, in Wales we have biodiversity net benefit, which hasn't really started, but in England it's a few years older, biodiversity net gain, where they have to compensate if they destroy 1 hectare. Yeah. They have to create 3 hectares of the wild habitat. So that's what the incentivization for here was. So they spread. So they paid, the developer paid.
That's great. And based on the arguments developed based on the wax cap studies. So you were able to make the case that this is a valuable topsoil that needs saving and translocation, which in my perspective, in our corner of Europe will be highly novel. I've never heard about that. Also, it's
they use the term irreplaceable habitat. That's the word they use for these things. I'm not familiar with that planning policy. I'm becoming familiar. But it's irreplaceable habitat, and then the developer has to compensate, if you like, compensate the natural world by creating new habitat in somewhere else. It's quite good, it's quite good for this. Some people ask me with the translocation, I think, like I said, it's an experimental method. It'll take many decades to find out how effective it is. So it shouldn't be done lightly. But if it's a hospital or wax caps, I think the sick people win every time. So the turf with these are being destroyed or it was getting moved.
No, it's a super great example. Thanks for it.
Thank you. There's a question from Isabel.
Am I allowed to ask a question, Elsie?
Yes, of course, of course.
I'm curious. I would like to ask a question actually to Lucy. Lucy, because this is a process that we will be starting within our specialist group, and I wanted to ask if you could expand a little bit. So when you started this process for the red listing, and you were talking at some stage about a prioritization of species, can you maybe briefly explain a bit how these prioritization steps occurred? Because I find this very interesting to apply, because we also have more than 6,000 species to red list, and we need to start somewhere. And having information from other colleagues on how they prioritize species is very important. Thank you.
Well, in our case, it was mostly based on what we know about the species, because we had a lot of species that we know that they were present in the Czech Republic, but we didn't know anything about them. A lot of Cartinario species, a lot of Entoloma species, a lot of Inocybe species, and such. So we really didn't want it to put much effort in gathering all the data, because searching the literature for those species, because we knew that we will end up with that data deficient category anyway. So it was this was the major reason for and also the species that was more known generally, but it's hard because you need to evaluate. In the end, you need to evaluate everything. So, and those low priority species were put into the red list just mostly because we want to highlight them.
Mm-hmm.
Yeah.
Thank you. Thank you very much.
Thank you. So I see, Gareth, you have a question as well.
But now that we can ask, the speakers can ask questions, I want to ask Isabel. I've always been fascinated with the aquatic fungi, or the aeroaquatic ones, how they always go down the stream, but then they have to have they often, or it seems some of them have an airborne state to blow them back up to the top. Does your surveying of the distribution enlighten us on where they might be? I think some might be endophytes in different plants.
Yeah, so some of the species have been described as endophyte of plants, so it's possible that just they probably spend some part of their life cycle as endophytes and another part of their life cycle as saprophytes. And this is actually still lacking a bit of empirical proof, but one of the things that people hypothesize is that this change between being an endophyte or a saprophyte might have to do not only with availability of resources, for instance, in temperate regions when the leaves fall into the streams, and of course you have more carbon available, but might also have to do with stress conditions. So for instance, we have a lot of the small headwaters that actually dry over a certain period of the year, and this might be a way for them to find the refuge during these times where there is no water, and then they can still survive as an endophyte, for instance. So yeah, but this is more hypothesized than actually empirically proved. So we need more information to really have this well established. Yeah.
Thank you.
Yeah, comes.
So then I can also have or let myself ask some small question also to Isabel, and a bit related to what Gareth just asked, because based on the case study you've shown, it's like, anyway, it's great to see that it's like not only like the diversity is within the protected area, but I was wondering whether there is like significant potential of like sampling bias or something that will give this like things that are not overlapping. Like I think you mentioned that there are at least 5, in this case study, 5 species, right, found outside that are like not there. So it could be like it was just overlooked, or it's just like something that there are other parameters that are like obviously affecting distribution of given species. But it's anyway great case study, and I'm looking forward for other like cases around the world, like to see how this,
Yeah, so for this particular case study, we only have for now the data analyzed for one-time sampling. So this is a snapshot, right? In other with other data that we actually compiled based on DNA, for some of these locations, we actually have temporal sampling. And I think this will be very important for us to also better understand if it was just randomly that we found this species outside and we didn't find it inside, but maybe, you know, if we sample it in a different time of the year, we also find it other way around, right? Either inside or outside. So I think the temporal question here is very, very important. And we are still waiting to analyze this data based on DNA to have these answers. So I think this will be coming in the next months, hopefully until the end of the year. But this is a really important question. So right now, we still don't have enough data to fully confidently say that these species only occur there. Now, based on this data, that's what it seems, but we need to, of course, be a bit careful here, and probably the temporal data will help us to complete the picture here. But it's still very interesting. And then when you're talking about which kind of factors may drive here these differences, one of the things that at least for aquatic hyphomycetes, then maybe not for all the species, but this is the group that I work mostly, that we have found is that sometimes streams that have a bit more nutrients kind of stimulate the species numbers. So for instance, one thing that happened in the regions that I have been studying in Portugal is that some of the streams that are very, very oligotrophic, so this means that they are very, very poor in nutrients, they actually can limit the number of species that we find in a given location. So if a bit further downstream and, you know, protected areas, they can be really pristine locations with really low nutrient concentrations. This can also limit the amount of species that we can have in a given location. So sometimes it's possible that in other streams where we have a bit more nutrients, I'm not talking about fully eutrophied locations, of course, because there usually we see a big decrease on the number of species, but a bit more nutrients availability can somehow stimulate the species also to coexist there. So there is this kind of bell shape trend where species tend to occur, and this is only for nutrients. And then we have all the other abiotic factors that we still want to explore to really understand what is driving this distribution of the species. Yeah, but thank you for your question.
Yeah, thank you. Yeah, because it gives me like a bit of the positive feeling that, oh, there's like still like some, because it's like largely an explored topic. So I was like, yeah, maybe it's like, because we did not care, I would say, as a humanity so much for either researching deeply aquatic fungi, and then obviously conservation is another level of unexplored, like big thing to do, and it's a lot of like difficult job. So I was like feeling that maybe there's like we have still a bit more time, because there's some, like, I don't want to say refuge places for some, let's say, species, but it's very interesting. So thanks a lot for your answer. And I think Alfred is having another question, so I will let you speak now so you can ask.
Thank you. Yeah, if it's allowed, I have another question for Gareth. I was going to ask if, or roughly how far are we, or what are the main barriers to having environmental DNA sampling as part of either the screening process or the environmental impact assessment process for forestry projects? Because a lot of the time now in Scotland, the sort of pre-planting process tends to be a botanist report, maybe an ornithology report, and there's this very fungi is sort of an afterthought. One of the slides you mentioned that eDNA maybe showed there was 6 or 4 or something extra species, but essentially I'm asking, is it valid enough scientifically to say this is a pretty good indicator of the active species in the soil? And sort of, yeah, basically how far are we to getting somewhere like that?
It seems to vary a lot depending on the council authority, because then you have the ecology, the planning process goes through the council ecologist, and if they say or suspect it might be what they might call ancient or semi-natural grass, then they can ask for a fungal report. And because the eDNA is a bit quicker than having to wait till the autumn to do a fruit body survey, people approach us and we do surveys on that basis. But it seems to be concentrated in certain council areas in Wales and not in others. So it depends on, I don't know how, I'd like to push the process further, but I don't know how. So I guess it's a matter of raising awareness with the broader plant ecologists, I guess.
Is it as legitimate as a study of the sort of visible fruiting bodies?
Well, what I suggest that people do, if they do eDNA first, say about now, they have the data, essentially they have a species list that's ready for the autumn, and then the field mycologist can go out and do the survey of fruit bodies later. But there's a problem that there aren't many people sufficiently skilled to be able to identify these grassland fungi. There's maybe in the UK, like top level people, maybe a dozen people who are really good. So they're limited, and they only have like 5-6 weeks to do the work in. So it should be the eDNA should make that process simpler.
Okay, thank you very much.
Welcome.
So if there are any more questions, I think it would be last moment to ask them. And I would like to thank you all for joining us today, because as Glenn already said, we were during the meeting in Uppsala a bit inspired, and for me it was very, very good to know that there are so many people and groups that are really motivated, and it's quite challenging to either work on red list or to work on a species to make it visible for conservation. So I'm glad we can have it during the FunDive webinar, and that it will actually land on YouTube. So it's going to be there for other people to see and listen at some point in the future as well. So I'm very grateful for that, and that we managed to meet and to have this meeting and discussions. So Glenn, I don't know if you want to say something, and if still there is one more question, we have some minutes, I think.
Yes, actually, I do want to say something more. What also came out of ECCF, or what is important, the next big important thing on the fungal conservation agenda is that there is now a new global strategy for fungal conservation. I won't talk too much about it because Greg Muller will give free webinars on that. So first one is tomorrow, actually, already, and then the other ones are shortly after that, in a week or two. So please all, for those interested, and I guess that will be most of you, please join those as well. And then maybe say again that this recording, and so also the image of this of this poster, if you have not seen it yet on your social media or on your email, you can also just look at it again on the recording, which will be on the FunDive YouTube channel in a day or two. Yeah, so that's it for me, I guess. I do have some questions more for Gareth, mostly, but I will write him an email.
Okay, cool.
So if there's not any more questions from the people in the chat, then I guess we can conclude for tonight. It's already 9:30, but it's still sun out, at least in Belgium. So I'll have a nice walk home. And then I thank everybody for joining, and again also to our speakers. So thanks everybody, and have a great evening.
Thank you for organizing.
Thank you, everybody. Bye-bye.
Thank you very much. Bye-bye.
Bye.
See you next month. Another webinar that we will hold.
Okay.
Definitely.
Bye.
Thank you. Bye.